Post-fertilization changes. After fertilization, the basal solan part of the oogonium separates from the trichogyne by the formation of a septum. The zygote increases in size and secretes a thick wall. The cell below and around the oogonium are stimulated and form a pseudoparenchymatous layer around the oogonium. The oogonium within its seed layer is called as spermocarp. Spermocarp is reddish brown in color. It is shed in water by degeneration of the basal cell and remain dormant throughout winter. Germination of spermocarp. On the return of favorable condition, the content of the spermocarp again turned green. The zygote nucleus divides meiotically. The first division of the zygote is by transverse wall, and subsequent divisions are at right angle to each other. Thus, 8 to 32 daughter cells are formed. Each cell metamorphoses into single biflagellated zoospores. Zoospores are liberated by breaking of the spermocarp and the zygote wall into two halves. They swim freely for some time, they settle down and eventually develop into new thalli. The alternation of generation. A distinct alternation of generation is not found in Coleochaete. The haploid thallus bear male and the female gametes on the erect branch which fuses to form diploid zygote. The zygote nucleus divide meiotically forming 8 to 32 haploid zoospores. Zoospores on germination give rise to haploid thalli. Thus the diploid phase is represented only by the zygote. Such a life cycle where diploid phase is restricted only to zygote is known as haplontic type. Affinity of coleochaete. The photosynthetic pigment chlorophyll A and B are the reserve foods. Starch are the reserve food material. Non-jacketed unicellular sex organs and the haplontic life cycle are typically chlorophysin feature of coleochaete. It shows resemblance with the member of the order Chaetophorales in its cell structure and the heterotrichous thallus organization. However, elaborate oogamous reproduction and retention of zygote on the haploid plant, presence of protective layer of the vegetative cell around the mature zygote, elaborate post-fertilization development of the spermocarp are some of the marked advanced features exhibited by the coleochaete. The occurrence of the discoid thallus in some species of coleochaete remind one of the character of the flat thalloid gametophyte of the liverwort such as Rixia. Brower in 1908 compared the spermocarp of coleochaete to a simple or primitive sporophyte of the early land plant. The more recent studies of Markensia and Pickett-Hepp in 1973, Graham and McBride in 1979 have pointed out that the coleochaete possesses certain features like the presence of phragmoplast during cell division, presence of glycolate oxidase and peroxisomes enzyme and the parenchyma in some species, which indicates its affinity with the early land plant. Hence, Stewart and Mathos in 1973 suggested that the land plant might have originated from coleochaete-like ancestor. There are remarkable analogies between the sexual reproduction of coleochaete and Nemoleonales among the red algae. The structure of oogonium of coleochaete markedly resembles the carpogonium of Rhodophyceae. Furthermore, in the coleochaete, the oospore is retained within the oogonium and the development of seed around it form a sort of fructification spermocarp, which is analogous to the spermocarp cystocarp of the Batrachospermum. Thank you.